In March 2026, social media erupted over an image beamed down from the International Space Station. The source was impeccable. Don Pettit, a veteran NASA astronaut commanding Expedition 72. He posted a photo of an eerie purple entity covered in chaotic, twisting tentacles, floating inside the pressurized cabin. The internet immediately panicked. Commenters compared it to the parasitic mimics from the Alien franchise, demanding NASA kill it with fire. The reality was completely harmless. That alien monster was a potato. Pettit had nicknamed it Sputnik 1. He was growing a purple potato variety in his free time, using a makeshift terrarium and a bit of Velcro to keep it from floating away. While millions of people were terrified by the appearance of a common vegetable safely secured inside the station, they were entirely distracted from a much stranger biological reality taking place on the outside. The extreme conditions of low Earth orbit exert immense selective pressure on biology. The environment up there actively forces terrestrial organisms to undergo rapid, bizarre mutations simply to survive. The potato's strained, tentacled appearance was the direct result of removing gravity from the equation of its growth. This diagram shows a cross-section of a plant root. On Earth, roots know which way is down because of specialized cells containing statoliths. These are heavy, dense particles that sink to the bottom of the cell. Their exact position acts as a mechanical cue, directing a steady flow of growth hormones downward to form an orderly root system. In the microgravity of the ISS, that sedimentation fails. The statoliths float aimlessly. Without a downward vector, the growth hormones diffuse randomly throughout the plant tissues. The potato's sprouts, those creepy tentacles, simply expand in all directions at once, desperately searching for a physical cue that doesn't exist. The harsh environment altered the potato in other ways too. The station's intense LED grow lights triggered a stress response, causing the plant to flood its cells with dark purple anthocyanin pigments for photoprotection. And without dirt to abrade its surface during a hydroponic growth cycle, the skin developed an eerie, glass-like smoothness. Removing the ancient constant of gravity forces a total reorganization of a complex organism's physical architecture. But while the potato enjoyed the pressurized, temperature-controlled interior, biology on the exterior hull faces a far deadlier threat: constant bombardment of galactic cosmic rays. To find out what survives out there, NASA runs the ISS External Microorganisms Experiment. Astronauts like Butch Wilmore conduct spacewalks specifically to swab the station's life support vents, hunting for microbial hitchhikers. This chart shows the radial growth rate of 2 identical strains of the common mold Penicillium rubens over 11 days. The control strain on Earth expands slowly, but the strain recovered from the ISS exterior balloons in size, growing nearly 4 times larger and over twice as fast as its terrestrial counterpart. It achieves this extreme resilience by rapidly producing a dense layer of melanin pigmentation. This dark biological armor absorbs heavy ions and shields the mold's DNA, preventing lethal double-strand breaks. The lethal cosmic radiation of low Earth orbit acts as an evolutionary pressure cooker, forcing common household molds to mutate into hyper-resilient organisms in a matter of days. In the vacuum of space, these adapted microbes have shifted from passive survival to aggressive consumption. Certain extremophile fungi use that same protective melanin to harvest ionizing radiation. Through a process called radiotrophy, they convert deadly cosmic rays directly into metabolic energy. We know this because the primary radiotrophic fungus currently being tested on the ISS, Cladosporium sphaerospermum, was originally discovered thriving inside the highly radioactive ruins of the Chernobyl nuclear power plant. Extreme environments on Earth are the training grounds for orbital survival. Scientists are now weaponizing these metabolic tricks. In the bioasteroid experiment, space-adapted microbes are deployed onto meteorite fragments to act rare metals like palladium, breaking down rock in zero gravity. The hostility of space unlocks latent metabolic abilities in these microbes, turning them from simple terrestrial molds into highly efficient biological machines. As space agencies prepare for long-duration missions to the Moon and Mars, the luxury of constant resupply ships from Earth will vanish. Astronauts will have to bring their own ecosystems with them into deep space, relying entirely on terrestrial biology for food, oxygen, and waste recycling. The biology we pack for these journeys will rapidly adapt to the cosmos. As we saw with the potato and the external fungi, we are actively forging entirely new biological phenotypes the moment we leave the atmosphere. The highly pigmented, tentacled organisms that emerge in orbit are exactly what we need. They will serve as our self-replicating radiation shields, new orbital biominers, and high-yield space crops. The true aliens we encounter in deep space won't be discovered haggard under Martian rocks. They will be the radically evolved versions of the life we brought with us.